had to run my Zoom for years. My phone. It's recording yourself? Huh? You're recording yourself? Okay, that's good. And you were posted on my. I mean, all four of them were there on. They were all on the Zoom meeting, so. Oh no, Candy was there.
What is going on now? Oh, I didn't take my attendance. Oh, shoot. Sanitize, please. Ugh.
Maybe they're on fire. Oh, Michelle, go.
What? Why can't I move this stupid thing?
I forgot where I was.
Oh, I'm so confused. Why can't I open this over here?
Just check. No, I'm scared to go out because I'm scared to go outside.
Oh, there we go. Sick. OMG.
Is the whiteboard wrong? Yeah, the whiteboard.
Wait, no. Flip it.
The whiteboard.
No, you two should be up here. Pretty sure.
What's the whiteboard? Which one?
This is the whiteboard, the main whiteboard.
Like this. Like this. This is natural, right? Like put the paper down. You put it like this. So the whiteboard is there. Why would the whiteboard be there? That makes sense. And you got to be like. Yep, I guess you're there. I didn't know where this was when I made this. I didn't know how close people were. We could probably just shuffle it back later. But so then next should be Michelle is there, behind Amanda, Wei Lim, Jay behind Yushan, yeah, Miguel, Thomas. Yes, 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 yes. Wait, Richard, what are you doing? Richard, you're here. You're beside Rudra. just to be clear, what you just did defeated the whole purpose of having a seating chart. Where is the seating chart? Where is it here? Let's touch all these things. Here? Do I sit here? Here? Just rub all my stuff on everything.
It's fine.
It's cool. It's cool. I'm cool. We're cool. It's cool. Okay. I don't know if these guys are here or not. If someone wants to like have their phone out and like tech support with them, let me know if something's going on. In theory, the meeting has started and Michael should be in it, but I don't see him yet, which is exactly what I said. Don't waste my time with this. Oh yeah, true. Is it today?
Yeah.
Today. Then Rudra. Blame Rudra. He promised.
You think his Wi-Fi is gonna?
He promised, man. He said Wi-Fi got so much better.
He
Kind
promised
sir,
Wi-Fi doesn't keep.
noble sir, something.
Noble.
Oh, he's sent like 3 hours of audio. Yeah.
Where's the seating chart? Why are you so close to it? So close to it. Stop.
Anyone else? Amanda's gonna put her viruses on them and pass them on so that everyone gets a little bit. Again, completely defeating the purpose. Okay, so let's see. I don't know if this works. We'll just try it. So in this case, I don't share screen, right? Or do I? No, I don't share screen. No, I don't need to. Yeah. I just want this to be recording. This is separate. They can see that. Bam, blam, blam. Hey, social distance.
Sorry.
Excessive social distance for you. Okay, and that's there. This here. On. Now, is this gonna work? I don't know. All right, people. We move on. We move on as though nothing happened. Yes?
Yes.
Because what the hell else are we gonna do, right? Sit here and be like, oh yeah, what did you do? Oh, that's fine. We're away. How did you feel? What does it make you feel that we lost all this? Oh shoot, I didn't write that again.
My phone is so bad.
Okay, we'll talk about our feelings and all this stuff.
It's hard to hold
No, we have been pushing forward, so we continue pushing forward. What did it say? You'll sleep when you're dead, right? We will sleep once the curriculum is over, like me and Mr. Euban, right? Mr. Euban? Yeah, high five. Boom. Staying with the sequence, man. We'll just go. So we'll finish this digestion. We'll finish 11.2. Have a test. Rest a little. Do like 3 more chapters. Then get ready for our fake end of year, and that's the time we can show. That is our reward. We get like 4 weeks to just reward. Yeah. Okay. Hang out, revise, go back over all the stuff that you may or may not have actually learned. See where we're at leading into summer and then into real end of year in the fall. Everyone, okay? We're just going to pick up where we left off, so we continue on to 6.1. I'm supposed to print something for you, and I forgot. I haven't had to print anything in 3 months. Here's the part that's tricky for me. You see what's happening? See, like I'm looking at that, which is just me. Now I want to do stuff here. Because I gotta like go here, like do this. It is very irritating. Okay. What is this? Why am I here? I want to If you want to download, you can do this on your own. If you want to open up, there's a file for drawing villi. I'm going to just finish in the week 14 section of e-learning and just use it till the end of this week, and then we'll go back to the normal iLearn, okay? Just because we're in the middle of it, and I don't want to like jump around too much. For God's sake, why? Well, it's because I didn't change it. Okay, so here we are, lesson 27. This file, I should have printed it for you all. So we're going to do the villus today, all right, which again, hopefully is mainly same stuff from from IGCSE, okay? We'll look at absorption. How is the villus adapted for absorption? That's mainly it. That's really the main part of today's lesson. Okay. Hopefully we finish this in like one more lesson where we do the only kind of really new part to this, but it's not all that new. If you, does this work? Yes. Come on. Not that new if you are in HL, because it's very similar to something that happens in the kidney. Okay. First of all, pretty much the only reason that was going to be great about coming back was what? For us, the science. Do all the labs that I said. If we were at school, we would. If we were at school, we'd do. If we were in class, we would. Well, guess what? We still can't. It kind of sucks. But we deal with it, okay? We would have probably gotten the microscopes out if I'm feeling a little bit more organized. I did try to get the microscope set up later this week in this room, so we look at these tissues. So what we do is we would look under the microscope at a section of the ileum. The ileum is what structure in normal words?
Small intestine.
Small intestine. Is it the beginning, middle, or end of the small intestine? The what? The middle, right, the middle. The kind of the majority of the small intestine, okay? If you see this, on this diagram, this is a drawing, not a microscope diagram, but I want you to note these 1, 2, 3, 4, 5 labels that you need to know, only the ones that are underlined, okay? So going from in to out, inside, this is, what do we call this area, this space? Of any biological structure that is tube-like. The inside is called lumen, correct. So the lumen of the small intestine, where all your food is being moved through. Then you have villus, okay, the villi, which are your folds, which we will look at shortly. So that's your first layer, right, made up of epithelial cells, which you'll see. Then you have some muscular layers that you need to know. So why, or what do those muscular layers do? Recall Monday's lesson. What do those muscular layers do? Help move the food by peristalsis. Thank you. Sorry, I didn't touch it. Nobody's not. Why? Hand sanitizer. Put on her shirt. Like what happens? I have no skin by the end. Oh, I actually did that. That was fun. Okay, the muscle layers help with peristalsis. Yes, so we normally attribute peristalsis to the esophagus. Excuse me. But of course, it happens throughout the digestive system, especially in the small intestine. Otherwise, your food would just sit there, right? Well, what are you waiting for? More food to come push it along? That'd be crazy. Then your whole thing would be just shoved with food. Gross. It'd just be like poof all the way through one long thing. Nasty. Okay? No, it's gotta move. It's gotta move in like groups, like little like ball form, or a chime is more like a mash, right? That pushes along the small intestine. So yes, ideally you'd be able to do a quick sketch. Let me just check something here. Yes, okay. So same labels, all right, but in a microscope image. This is what it would look like in real life. Can someone tell me how come these pieces are like floating in midair? How come it's like islands of tissue? Why is that the case?
Social distancing.
Sorry? It's cut. Something about the cut, yeah. What are we actually looking at there? What are those little pieces? You got to be like a bit of a spatial orientation person. What should that look like in the ileum if you're looking under a microscope at not a slice, but if you're looking at the tissue? What are these things? Well, it says it right there. Villi, right? What do villi actually look like? Kind of. Right? What do we use to draw it in IGCSE? How did we draw villi in our notebook? With your fingers, right? So they kind of looks like these things. So then how come these are like all separated and disjointed? Nice shot. No. IGZLI is a good idea, but no. I mean, you have to think of it in like 3 dimensions, right? If you have villi like this, and you like they're in 3D, right? So there's many of them, like all throughout the thing. If I take a slice across the ileum, right? This is not going to be like perfect and like right across the flat part, and you get a perfect flat villus like this. No, some of them are going to be folded. So when I cut it, bam, there's going to be pieces of this one, pieces of that one, kind of all different places, right? They're all going to be like interacting like this. So if I take a slice, it's not going to get you one very nice round villus. So that's why you have kind of a different view here. So this part we know, when we draw it, it looks more like, we'll see, get to this in a second. It looks more like this is how we draw it, but in real life it's like 3 dimensions, right? And like, this is very rare to get a picture in this plane. So what do those layers that we labeled do? You have, first let's start with what we know, the epithelial cells. So those are the very, very outer layer, right? The single cells that make up sort of the boundary between inside the body system, the blood, and the lumen. And then you have these muscular layers. So you've got first some connective tissue. You've got mucosa and submucosa. I guess let's go from inside to out. So mucosa is like just like a connective tissue layer, okay, which is part of the villus structure, if you will. So like mucosa plus epithelial cells equals 1 villus. Then you have connective tissue. This is linking your villi to the muscles. So then the next part, notice, remember, circular and longitudinal, the 2 layers of muscle, same as in the esophagus. Right? So the circular and longitudinal makes up the muscular layer. So you should be, again, familiar with these layers, right? Mucosa, submucosa, muscle layer, sclerosing. I don't know if anyone understood what I was trying to say last night, but I have HL for the other group today, which is Janaki and William. So I'm actually just zooming from here to them. What a trip, man. I don't have period 1, so I don't have a period 7. HO. I don't stand here. This video is going to be extremely boring. Because I'm recording the lesson with those guys. They just talk like this. This is why I said this is, I don't think this is the right choice. It's very weird. I'm not editing that either, forget that. And like if I don't do anything on the board. Oh, we'll see. Okay, good. This is kind of new, right? We never really talked about the different layers of the intestine. So that will be new for you. All right, just as a preview before we move ahead, what are those two words with A that this whole topic is about? Starts with letter A. Good. Absorption and assimilation, right? Absorption is really like if you had one word to describe what the small intestine is for, what the ileum is for, absorption, right? Bringing the nutrients that have now already been digested or are just getting digested a little bit along the way on the cells of the villi, but basically they're digested, they're small enough, they need to be absorbed into the bloodstream. Later, we'll deal with assimilation. How do we build new molecules out of them, out of the stuff that has been absorbed? So what we'll look at now is, okay, this is done. This is what I just said. First, digestion, then absorb, then assimilate. Step 1, step 2, step 3. Right? When you eat food, it's too big to absorb, so you have to break it down. Once it's broken down, it can be brought into the bloodstream. Once it's in the bloodstream, it will go to various parts of your body, different cells all throughout, so that those cells can build structures of their own. They can build their own proteins, their own fats, their own carbohydrates, make new cells, divide, do all the things that you need your cells to do for you. All right, so this yellow box really summarizes what you need, along with vitamins and minerals. So we don't mention these often, but those are included in the absorption. The main ones we talk about are monosaccharides, which are like sugars, right? The breakdown products of carbohydrates, amino acids, and then the two parts for fats. They are absorbed by the villi. Yeah, usually this lesson would be in microscopes, that's why. Okay, if you're done early, you can start sketching out your villus to label and annotate. So as usual, we'll do one of these. I'll just draw one. Maybe I'll do one more. So that I have some space. Can we talk about, did you guys watch that guy that drew the thing? I think I sent it on IG that like, he draws like the sports things on the whiteboard. There's so many questions. Like one, how do you get so many colors of whiteboard marker? That's the main question. Everyone okay if I move ahead? Does anyone still need this? Good. Good. Satisfied? Okay. Ooh, no. I don't want to give you that yet. Not yet, man. Get carried away here. Alright. Can we make a quick list on the side before we even annotate? What do we remember are the adaptations that the villus plus the villi, remember villus is one, villi is multiple, that the villi have and make it good at absorption. Good at absorption. What are some of the features? Surface area because of folding. Right? All these finger-like things means you have high surface area, more places for absorption to take place. Fantastic. I'm just going to make a list here, and then we will actually put it over there on the drawing. What else? Short distance. Because why? What allows for the short distance? Correct. One cell thick wall. What is something to be careful of when saying this part? Don't say what? Where can someone make a mistake saying this part here? One cell wall thick. Why is that wrong? Yes, but scientifically speaking, why is saying cell wall not a good idea?
It's only for plants.
Thank you. It's simple. It's easier than you guys are making. Cell walls are in plants, right? Animals don't have cell walls in their cells. So if you say it has like one thick or like thin cell wall, that's usually how it goes. People say thin cell wall. No. Thin wall of the villus, right? This is the wall of the villus. This is the barrier between outside and inside. So the wall of the villus is thin, but it is made of a single cell layer. When you're talking about this, do not put cell and wall right beside each other. Then all of a sudden you're talking about plant cells. Okay, there's probably more. No, there are definitely more.
Constant gradient.
Good. Concentrate, maintain. Let's say maintain. Concentration gradient. How? Good. Blood, because constant blood flow. Another thing for short distance, actually, something else that's related to short distance. What are they called? Capillaries, right? Dense capillary. What's the word we usually add? Dense capillary network. Right? Basically meaning the capillaries are very close by. They're right behind the wall. Okay? Makes the diffusion pathway short. Trying to think, is there something more? Oh, yes, forgot another one. What's another thing that allows for surface area? Microvilli. Okay, we'll draw that in a second. And one more, what's the green thing?
Lacteal.
Lacteal, right? Absorption of fats. Not fats, fatty acids. Okay, now let's label this thing. So, did we get everything? Many villi, increased surface area, single layer, okay? So you can avoid the whole wall problem by just saying single layer. So just don't say wall at all. Single layer of epithelial cells, microvilli on every single cell, lacteals, capillaries, bam, nailed it. Unbelievable. Who says e-learning doesn't work, right? So smart. I'm just gonna add this. Obviously, you wouldn't see this in this situation. Let's do this then. Like, bam, one of these things. It's been so long since I used a whiteboard marker, guys. That's terrible. Okay, this will come into play later when we discuss how things actually go across the epithelial wall into the bloodstream. We're going to talk about the transport of different types of nutrients because they actually move in different ways, okay, from outside to in. Microvilli, I'm just going to add labels here, okay? Capillary network. Oh. Lacteo. And I think that's about it. Yeah, you might want to just indicate like blood flow. The reason we do red and blue here is to show sort of this, that there's a directional flow of the blood, that it's constantly being replenished, right? Otherwise, this is, in real life, that's highly inaccurate to draw it like that, but whatever. But it shows, like what Richard says, like new blood is always coming, so that new blood would be lower in nutrients, so it's more likely that the nutrients from out here, right? This is high, no, let's write the word. High nutrient, right? So this is monosaccharide, amino acids, blah, 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 is moving across a diffusion gradient, right? So if this is high and it's moving toward low, right? In here would be low. So it's always going to go that way, and then when the blood pumps away, we're assuming that food is. Right, we're assuming that food is flowing this way. All right. So as food is passing and bumping into all these villi, they are likely to be absorbed because of, what's the word? What's the answer to all questions? Diffusion, right? Diffusion. Don't forget about it.
Wait, then it's the blood like oxygenated for.
It's being shown that way because then it makes sense for these arrows to go this way. This might happen. Like you might say that, like, because like these capillaries may be giving blood to these cells to survive, right? And then becoming deoxygenated along the way. It's also possible that they just stay oxygenated and the oxygen goes to somewhere else. It's just a way of showing the direction of flow, actually, by just going red to blue. Okay, let me just check here. Oh yeah, we're gonna stop there. So the important thing to note is, one, into the bloodstream, we absorb amino acids and sugars, right? Maybe we should make a note of this. AAs plus monosaccharides, which are sugars. And maybe some FAs, fatty acids, and glycerol. Okay, some. Lacteals absorb only fatty acids and glycerol. Okay, so this is majority of fats are going to go here. The lacteal is part of the lymphatic system, right, which helps to build what kinds of cells does the lymphatic system? White blood cells. Yeah, white blood cells. We sort of touched on it in HL, I believe. Yeah. So you have the lymph nodes. White blood cells can like go in there. They collect extra fluid from that leaks out of capillaries in different places, and then yeah, you have networks of white blood cells. Good. Right. I think that's it for this part. I didn't want. This was intentional, guys. I know that probably most of your lessons are like what I was making fun of earlier, right? Let's get back.